Let us learn the structure of nucleus in this class. To make our understanding easy, we will learn nucleus under different heads like introduction, morphology, ultrastructure, and functions of the nucleus. Nucleus, the term nucleus is derived from a Latin word nux, which means nut or heart of the cell. Robert Brown discovered and named nucleus in the year 1833. He was a person who discovered and he named it as nucleus in 1833. It is considered to be the most essential of all the cell organelles, which commands every metabolic activity of the cell. That is why it is called the controlling center of the cell. Almost all DNA of the nucleus is contained, replicated, and transcribed in the nucleus. That is, the DNA is present in it, it replicates inside, and it transcribes inside the nucleus. Nucleus this controls different metabolic as well as the hereditary activities of the cell. The Greek synonym of the word nucleus is karyon. So, nucleus is also called as karyon. It is a main distinguishing feature of almost all the eukaryotic organisms. Morphology of the nucleus can be studied under different heads. The first one is the occurrence of the nucleus, the position, number of nuclei present inside a cell, the shape of the nucleus, and the size of the nucleus. A well-defined nucleus is present in almost all the eukaryotic cells, except in the mature mammalian erythrocytes and the sieve cells of higher plants. These two cells also have nucleus in their young conditions or during the process of development. The nucleus which was present in these cells, it disintegrates, and the mature cells do not show any nucleus. Prokaryotic cells do not have a true nuclei. Instead, the nuclear material is called genophore or a nucleoid. It is either called a genophore or it is called a nucleoid. The position of the nucleus within the cell. The position of the nucleus is a characteristic feature of the cell, and it is often variable depending on the cell type. Usually located in the center, but is pushed towards one side as the cells start differentiating. In glandular cells, the nucleus remains at the basal part of the cell. In mature cells, the peripheral region, you find the nucleus. This is because there is a large vacuole in the center of the cell. So if we want to draw every cell, suppose this is a cell, usual position is the center part of the cell. In some cells like the glandular cells, the basal cell has the nucleus on which the gland might be present. In some cells like the parenchyma, mature parenchyma cells, we draw the cells. This is an outer cell wall. Imagine this to be a parenchymatous cell. Because of the presence of a large central vacuole, the nucleus is pushed towards one periphery. So it is not constant in all the cells. Depending on the cell type, the nucleus has different positions. The number of nuclei also varies from cell to cell. Usually we have cells which have a single nucleus. Such cells are called mononucleate cells. But we also have cells which do not have a nucleus. These cells which do not have nucleus are called the anucleate cells. The examples are the RBCs and the sieve cells of phloem tissue. These are mononucleate cells, very common in almost all the cells of the plant, which have a single nucleus. This is the diagram which represents a mononucleate cell. There are few cells which have two nuclei in a single cell. These cells are called binucleate cells. The example is the tapetal cells of Euphobiaceae are binucleate cells. There are certain plants which are multinucleate or xenocytic in their condition. That is, they have many nuclei per cell. Every cell will have more than 2 nuclei. The examples are the endosperm of the seeds and Syncytium fungi. This multinucleate condition of the cell is due to a free nuclear division. That is, when the nucleus divides, the division of the nucleus is usually followed by a cell wall. Here, instead of formation of cell wall, the nucleus just keeps on dividing without forming a cell wall, as a result of which the cell accumulates large number of nuclei, resulting in the formation of a multinucleate condition or a xenocytic condition. So the number of nuclei is also not constant in all the cells. It differs from cell to cell. The shape of the nuclei. Usually, the cells have a spherical nuclei which is present within their cell. It is usually in the center. But the shape also varies. In cells which are spheroidal, cuboid, or polyhedral, the shape of the nucleus is spheroidal nucleus. That is, if the cell is spherical, cuboidal, or polyhedral, the cells will have spheroid nucleus. If the cells are cylindrical, prismatic, or fusiform, they have a ellipsoidal nuclei. The squamous epithelium of animal cells have discoid nuclei. The leukocytes have a horseshoe or a trilobed nuclei. This is the example of a leukocyte which has a trilobed nuclei or a horseshoe shape of nucleus. So in some plants and some cells, if they are spherical or polyhedral, the nucleus is spherical. If the cells are cylindrical, they have ellipsoidal nuclei. Or if they are fusiform, even then they have a ellipsoidal nuclei. So the shape of the nucleus is not constant in all the cells. It differs from cell to cell. Also differs based on the type of cell in which it is seen. The size of the nuclei. Generally, the nucleus occupies 10% of the total volume of the cell. The size varies between 5 micrometers to 25 micrometers in diameter. It is correlated to the number of chromosomes. That is, it is directly proportional to the number of chromosomes. More the number of chromosomes, the bigger the size. The cells having triploid or tetraploid chromosomes have a large nuclei when compared to those of diploid or haploid set of cells, nuclei. The relation between the nuclear volume and the volume of the cytoplasm is expressed numerically and is called the Nucleoplasm index, which is represented by NP. NP can be calculated as Vn by Vc minus Vn, where Vn is the volume of the nuclei and the volume of Vc is the volume of the cytoplasm. Thus, the size of the nucleus depends on the volume of the cytoplasm, the number of chromosomes, and the metabolic phase of the cell. The metabolic phase of the cell is during cell division. If we take the example of cell division, during the initial cell stage, division stage, that is in prophase. In prophase, the nucleus is very large it enlarges because of accumulation of water. And the same thing if we see in telophase, the nuclear size is slightly smaller when compared to this. So during the division stage also it differs. Not only during the metabolic phase, in this division stage also it differs in plants. So the size of the nucleus is not constant in all the cells. It differs from cell to cell, ranging from 5 micrometers to 25 micrometers in diameter. Let us now learn about the ultrastructure of the nucleus. The ultrastructure of the nucleus can be studied under 4 different heads. That is, whatever is present in the nuclear structure divided into 4 different parts. The first one is the nuclear envelope, or it is also called as the karyotheca. The second one, the nucleoplasm, called as the nuclear sap, or it is also called the karyolymph. The third part is the chromatin or the chromatin network, and the last one is the nucleolus. So under these different heads, we can talk about the structure of the nucleus. This is a diagram which represents the nucleus. It has a double membrane outer layer which forms the nuclear envelope. We will talk about nuclear envelope in detail in the subsequent slides. Then inside the nuclear envelope is the nucleoplasm. Embedded within the nucleoplasm is the chromatin network, which appears as fine, thin, thread-like structures. Then inside, embedded within the nucleoplasm, that is the matrix which is present is the nucleoplasm. Embedded within the nucleoplasm is the nucleolus. So each part we will learn in detail. Let us talk about the nuclear envelope. The nuclear envelope is surrounded, the nucleus is surrounded by a membrane called the nuclear envelope, or it is also called as the karyotheca or the nuclear membrane. If we can draw the diagram like this of the nuclear envelope. The nuclear envelope of the nucleus. It is not continuous actually, so I'm drawing it like this. They have minute openings on the surface. This is the nuclear envelope. This is the outer membrane and this is the inner membrane. The outer membrane and this one is the inner membrane. Each membrane is a unit membrane. That is, they are made up of lipids towards inside. That is a bilipid layer and surrounded by the proteins. This is a unit membrane structure or a trilaminar structure, which is very common for all the membranes. So both of them, each one has this structure, unit membrane structure, and they have a thickness of 75 to 90 Armstrong units. Each membrane is 75 to 90 Armstrong units in its thickness, and it is made up of lipids and proteins. Each membrane is 75 to 90 Armstrong units in thickness and is lipoproteinous in nature. These two membranes are separated by a space, this gap empty space which is seen here. This is the space which is called the perinuclear space, and this space is around, the distance between the two membranes is about 100 to 150 Armstrong units. That is, both the membranes are away from each other by 100 to 150 Armstrong units. The outer membrane is rough. The outer membrane is rough due to the presence of, this is the nuclear membrane. It is a double membrane, the outer and the inner membrane. So this is the outer membrane. This is the inner membrane. Between the two is the space which is called the perinuclear space. The outer membrane is rough due to the presence of the ribosomes on it. The outer membrane is also in continuation at certain points with the endoplasmic reticulum. That is, we can see that the endoplasmic reticulum is attached to the outer membrane. And there are small gaps on the nuclear membrane. These gaps or these openings are the nuclear pores or nuclear apertures. We'll talk about nuclear pores later. The inner membrane is smooth. It's covered by a smooth layer, and this layer is fibrous in its nature and is made up of a protein called lamin. So this is also called as the nuclear lamina, and it supports the structure of the nucleus. This lamin structure supports the structure of the nucleus. We'll see the remaining part now. The same thing is written here, that each membrane is 75 to 90 angstroms. Then the space between them is 100 to 150 Armstrong units. The membrane is rough due to the presence of ribosomes, and the membrane is in continuation with the endoplasmic reticulum, which we have already seen. The inner membrane is coated with an electron dense layer, which is called the nuclear cortex or nuclear lamina, which is around 300 Armstrong units in its thickness. And we have also seen that the nuclear envelope has small openings which are called as the nuclear pores. These nuclear pores, the pore was first demonstrated by Callan and Tamlin in 1950 in the amphibian oocytes. The nuclear pore was first demonstrated by these two people in 1950 in the amphibian oocytes. The nuclear envelope is perforated by a number of pores or apertures. These apertures are called the nuclear pores. Just now we have seen that there are small openings on the surface of the nucleus or on the membrane, which are called nuclear pores. The membrane around these pores is in continuity with that to form a round lip-like structure. That is, the nuclear membrane is not a complete circle like this with two concentric membranes. It is not like this. So the membrane is something like this: outer membrane in continuation with the inner membrane, the outer membrane in continuation with the inner membrane. So wherever it folds, it forms a lip-like structure. This lip-like structure is formed. The number of pores per unit area differs with the type of physiological function the cell has or the physiological state of the cell. Then the pores may be around 100 or as many as 5 into 10 to the power of 7, that is every nucleus. Depending on the type of cells, the nucleus might have 100 pores or there may be as many as 5 into 10 to the power of 7. That means these number of pores may be many in some it may be only 100, in some it may be many. The diameter of the pore varies from 40 to 100 nanometers. This region, this is the diameter of the pore, which might be 40 nanometers in its diameter or it might be 100 nanometers in its diameter. The average is around 80 nanometers. The arrangement of pores on the nuclear membrane may be in rows, clusters, hexagonal patterns or random. I'll draw the nucleus like this for time being and imagine this to be the outer surface of the nucleus. The pores might be arranged like this in rows on the surface. It should not be imagined as a circle. It is a spheroidal structure. It might be arranged like this in the form of rows, or it may be in clusters arranged like this, or the pores might be in the form of hexagonal patches. The patches appear as hexagons, but there are pores in the pattern. Or in some nuclei, we find random arrangement of pores on the surface. There is no specific arrangement. So the arrangement of pores may be in rows, in clusters, or in hexagonal patterns, or they might be random. The nuclear pore is surrounded by a ring or a cylinder which is called the annulus. I'll draw the diagram now and explain it again. This annulus also was explained or observed by Callan and Tomlin. It extends both into the cytoplasm and the nucleoplasm. The annulus along with the nuclear pore forms the pore complex. So to make it very clear, this part of annulus and how the pore complex is formed, I'll take another screen and try to explain it on that. So we'll see this as, let us see the, imagine this to be the nuclear membrane. This is a membrane. I'm trying to draw to show you that there are two membranes. Imagine this to be the outer membrane, this to the inner membrane. The surface view of the pore will be something like this. The nuclear pore. There might be many nuclear pores on the surface. I'm drawing one here. So this ring-like structure which is present here is the annulus. Here also, if you see, this extends down between the two pores. You can see the pore is not single only on the external surface. This pore is a continuation. It appears, it embeds itself between the two membrane surfaces. So this is the vertical view. If you see, this is the vertical view. This pore is surrounded. If I with the help of a cylinder we can explain this, or if I draw a cylinder like this, imagine this part to be the outer surface and this part to be the inner surface, or outer membrane and inner membrane. On this side also, this is the outer membrane and the inner membrane. Which membrane is this? This is the nuclear membrane, the outer and the inner nuclear membrane. And this region is the pore region. The pore region, this whole thing is a pore, the aperture through which the transport of material occurs. The pore region has a ring-like structure on the outside and also towards inside. It slightly extends towards the cytoplasmic end. It extends towards the nucleoplasmic end. That is, a part of the annulus can be seen protruding out towards the cytoplasm, protruding in towards the nucleoplasm. This ring-like structure is called the annulus. The annulus along with the pore, the annulus plus pore, they form a complex structure which is called as the nuclear pore complex. And this pore complex is not simple as we see here. This annulus on the outer surface has small cylinders arranged on the periphery of that, or arranged on the rings. There are around 8 cylinders, like 1, 2, 3, 4, 5, 6, 7, 8. 8 cylinders which are arranged on the annulus. They are called cylinders, ovoids, microcylinders. These are towards the outside. In the same pattern, you have the annulus towards the inner side, on which again there are 8 microcylinders or ovoids which are arranged. Between the two, there is a central plug, that is here, somewhere here inside the pore, there is a central plug which is around 350 Armstrong units in its thickness. All these ovoids or microcylinders join to the central plug by cytoplasmic strands. They join together to the central plug by cytoplasmic strands. And this complete structure is explained by a number of biologists. Let us see the pore complex. This is what I had been explaining now. The annulus contains 8 subunits which are arranged radially in a radial symmetry. They occur in a set of 8. Here you can see there are 8 and there are 3 sets of 8 cylinders. 3 sets of 8, one set on the cytoplasmic end, one set on the nucleoplasmic end, and one in the center. So this is the outer cytoplasmic end, this is the nucleoplasmic end, and this is the central region. This is how the pore appears. The annulus along with these cylinders together and the pore together, they form a complex structure which is called the nuclear pore complex. The central cylinder, this is again another view of the same nuclear pore complex. The pore, it has a central cylinder which is 40 to 350 Armstrong units in its and is present in the center. And all these cylinders, they connect to the central cylinder by filaments, cytoplasmic filaments, which are called the axial filaments. These are the axial filaments which help in connecting the microcylinders to the central plug. The functions of a nuclear envelope. You can write a number of functions for nuclear envelope. The first one is it is an interface between the cytoplasm and the nucleoplasm. That is, it does not allow or it interferes or it inhibits the cytoplasm being mixed with the nucleoplasm. Both are kept separately because of the presence of the nuclear membrane, and this is very important because of a number of other reasons. We will come to those reasons now. It separates the genetic component from that of the protein machinery. What is protein machinery? The ribosomes and the enzymes which are required for protein synthesis. It provides protection to DNA against cytoplasmic enzymes. If the DNA comes out into the cytoplasm, there are all chances of DNA getting denatured or being broken down by the enzymes. So the DNA is protected inside the nuclear envelope. It helps in the transport of macromolecules and small organic material, water, ions across the membrane, or from the nucleus into the cytoplasm, or from the cytoplasm into the nucleus. It provides a surface for the attachment of structural elements of the cytoplasm, like myofibrils get attached, microtubules get attached, or microfilaments, they get attached to the nuclear envelope. The telomere or the centromeres of the chromosome may be attached to the envelope from inside. There are chances that lamina helps in attachment of the telomere or centromere of the chromosomes. The nuclear sap or the nuclear matrix. So the second part of the nucleus is the nuclear sap or the nuclear matrix. This is the material which is present inside the nuclear envelope. Inside the nuclear envelope, the material which is present is called the nuclear sap. This sap is homogeneous. It is granular and acidophilic in its nature, and is called nuclear sap, nuclear material, or karyolymph. It is rich in proteins, and it also has small quantities of DNA, RNA, and phospholipids. It is a site of active enzyme activity. That is, enzyme activity is more in the nuclear sap. So it contains, and thus it contains the DNA and RNA polymerases, ribonucleases, alkaline phosphatase, and dipeptidase enzymes. What is the function of the nuclear sap? The function of the nuclear sap is to help replication of DNA, transcription of RNA, transport of material, and the formation of spindle during the cell division. Replication of DNA is the DNA forming another DNA. Transcription of RNA is the DNA opens up and on one of the template the RNA is synthesized. And transport of material is, this is the nuclear sap which is present inside in the form of granular substance, and through the pores the material from outside, that is from the cytoplasm, it enters into the cell or it might go out of the nucleus, enters from the cell into the nucleus or from the nucleus into the cell. So the nuclear sap and nuclear matrix help in transport of materials and they also help in enzyme activity. The next one is the chromatin material. The chromatin material is nothing but the chromosomal matter which is present inside the nucleus embedded within the nucleoplasm. This chromatin material, it appears as fine, thin, thread-like structures which are distended into a network, or that is why it is also called as the chromatin reticulum or chromatin network. During cell division, they condense to form rod-like structures. If you see the interphase nucleus, the chromosomes are thin thread-like structures, hardly visible, very faintly visible. Then during metaphase, if you look at the chromosome or the chromatin material in metaphase, they appear as condensed material. They coil because of supercoiling. They become stout and thick and are very clearly visible as rod-like structures. And what is present in these chromatin material? It is the DNA, RNA, and proteins. These three make up the chromatin material. What are the types of proteins which are present in the chromatin material? There are two types, histone proteins and non-histone proteins. Both of them are seen in the chromatin matter. The proteins and the DNA, they exist in 1:1 ratio on the chromatin. The histone proteins are of 5 different types, or 5 classes of histone proteins are seen on the DNA. They are H2A, H2B, H3, H4 and H1. Of these, the first 4 of them, they have both acidic and basic amino acids. The non-histone proteins are concerned with gene expression. The example is the RNA polymerase. The chromatin which is present inside might be loosely coiled or it might be tightly coiled. The loosely coiled chromatin is light in color or lightly stained. Loosely coiled, lightly stained region. This part of the chromosome or the chromatin is called euchromatin. And this euchromatin has active genes. Whereas the darkly stained and tightly coiled region of the chromosome is the heterochromatin. This is euchromatin, this is heterochromatin. This has loosely arranged filament, this has coiled filament, this is lightly stained, this is darkly stained, this has active genes and this has inactive genes. Under electron microscope, the chromatin appears like a string of beads. When we look through a compound microscope, we can just see the chromatin material as threads like this. even when we are doing mitosis experiment and all, we can see threads. But under electron microscope, when we look at the chromatin material, it is not in the form of threads, but it appears as a string of beads like this. Each bead-like structure is a nucleosome. The nucleosome is a flat disc-shaped structure which is around 11 nanometers in diameter and 5.7 nanometers in its length. Now we will see what is this nucleosome. The nucleosome is formed, it has a core which is formed with the histone proteins and is coiled by the DNA. The histone proteins, like the H2A, we have H2A, 2 units of H2A, 2 units of H2B, and H3, 2 units, and H4, 2 units. There are altogether 8 of them which form the central core of the nucleosome. This core of the nucleosome is connected, or it is held with a clamp-like structure or a clamp-like part, which is the H1 histone protein. Around this core, imagine everything, all 8 things are here. Octamer, the octamer is here, and this is clamped by the H1 protein. This core is surrounded by the DNA. The DNA coils like this, one complete coil and another 3/4 coiling. In some of the books it is mentioned as 2 complete coils. This is the core which is surrounded by the DNA, coiled by the DNA, 1, and the next is 3/4. And then the DNA extends like this. Again it coils around another core, 1 complete coiling, another 3/4. Again it extends. There is another core of the nucleosome, 1 complete coil. So like this, it forms a structure. This octamer which is coiled with the DNA is the nucleosome. And this DNA which is joining the 2 nucleosomes is called the linker DNA. This linker DNA it has around 146 base pairs in it. So this is a nucleosome which appears as, under electron microscope, the chromosome appears like this. And this beaded structure is this part which is formed by histone proteins and the DNA which coils around it. The next part of the nucleus is the nucleolus, which is seen inside the nucleus. We'll briefly see the structure of the nucleolus. It is spherical in its nature, and it is formed near a nuclear organizer of the chromosome, and it is very dense in its nature. The nucleolus is a large spherical body which is present inside the nucleus. Usually only one nucleolus is present within the cell, within the nucleus, but in some cases more than one nucleolus are also found. It was first described by Fontana in 1781. So very prominent in the cells which have a high rate of protein synthesis. It is found closely associated with the nuclear organizer region of the chromosome. The number of nuclei varies in different cells. It may be only 1 in a haploid set of chromosome, or it may be as many as 200 nucleoli in the oocytes of amphibians, and they have active in RNA synthesis. They have active RNA synthesis. So this is the nucleolus. We want to draw the nucleolus. The structure, there is an ultrastructure of the nucleolus which we are going to discuss little later. The nucleolus is formed near a region of the chromosome which has the nuclear organizer. It is dense in its nature, and it is usually only one nucleolus per nucleus, but sometimes more than one is present. Here we have taken the example of 200 nucleoli. It actually disappears during cell division. During the process of cell division, the nucleolus, the nuclear membrane, everything disappears. And then it reappears at the end of cell division. Now let us see the structure of the nucleolus. The nucleolus has three important components, or it is formed by three important parts. And this is the structure under the electron microscope. The one of it is amorphous matrix. It is proteinaceous ground substance in which granules and fibers are suspended. This part, it is proteinaceous granular matrix in which the fibers and the granules are suspended. The second one is the granular zone, which consists of particles of 150 Armstrong units. These are ribonucleoproteins. They are the granules which are present towards the peripheral region. These granules are slightly bigger, ribonucleoproteins, and present towards the periphery, and they form the granular zone. They are around 150 Armstrong units in thickness. The last one is the fibrillar zone, which comprises of fine filaments of 5 to 10 nanometers in diameter and several hundred nanometers long. These are the fibrillar part or fibrillar structures, which are 5 to 10 nanometers in diameter but several hundred nanometers in their length. They are formed of rDNA, rRNA, and ribonucleoproteins. The functions of the nucleolus. Most active site of RNA synthesis. RNA is formed in the nucleolus and produces 70 to 90% of cellular RNA. It is related to biogenesis of ribosomes. That is synthesis of ribosomes. This includes transcription of rRNA, processing of periribosomal molecules, assembly of ribosomal units, and plays a very significant role in mitosis, disappearing and reappearing. Actually, it doesn't disappear completely. Only thing is the nucleolus material, which is in the form of a dense structure like this, all of them disperse. The fibrillar, the granular part disperse within the nucleus, and so it doesn't appear. It is not seen clearly. Later in telophase, they again assemble together to appear as the nucleolus. The last part is the functions of the nucleus. Here I have written only 3 functions of the nucleus, but the function of every part of the nucleus can be included into this. I broadly wrote metabolism. It is the controlling center of the cell, controls majority activities. It contains DNA, so plays an important role in heredity. It helps in synthesis of RNA, and that occurs in the nucleolus. Whatever we have studied under different parts of the nucleus, all the functions can be jotted down here. This is an additional information where we have seen about nuclear pore. We talked about the number of pores, the diameter of the pore, etc. The nuclear pore occupies 20 to 36% of the nuclear envelope. The number of pores in the nuclear membrane is correlated to the transcriptional activity of the cell. During mitosis, the lamina proteins. What is the lamina protein? In inner membrane is surrounded by a 300 nanometer thick layer, that is the lamina. It supports lamina protein. It becomes phosphorylated and nuclear envelope disassembles. It becomes phosphorylated, so during cell division the nuclear membrane will disappear. Each species has a characteristic content of DNA, which is constant in all individuals of that species, and this is called the C value. This is called the C value. In man, the 3 picograms of DNA per haploid genome is present, that is 3 into 109 base pairs 1 picogram is equal to 10 to the power of minus 12 grams. The nuclear organizer of the chromosome contains 18S, 28S rRNA genes. The nuclear lamina, it provides support to the nucleus and has special protein called lamin. We have discussed this. And it serves as a binding site for chromatin. This is just an additional information which you can add when you are doing the respective part of the nucleus. Wish you all good luck.